Okay, dears, let's talk about something about cell structure and its functions. You are quite familiar with the word cell. As you know, cell is the basic structural and functional unit of the cell. By the middle of 19th century, biologists had formulated cell theory, which is a fundamental concept in biology, according to which all organisms are composed of one or more cells, and the second postulate is new cells arise by the division of pre-existing cells, and then a cell is the basic structural and functional unit for all living organisms. So all these things became possible because of the discovery of the microscope, because cells are very small. We cannot observe cells with our naked eye. So to observe cell, we need a microscope, and with the emergence of microscope, we discover the cell and its content. And moreover, we use different techniques in which we use microscope and lenses and other things to observe more clearly what is inside the cell and how cells which are busy in making proteins do these jobs. So here are some techniques. In today's tutorial we will discuss the technique which is called the cell fractionation. And as you know, cells is composed of a cell membrane, and inside the cell membrane there is a liquid environment which is called the cytoplasm, which is inside the cell membrane and the nuclear membrane. Like this is the cytoplasm. You can see this is the cell, this is the cell membrane, and inside this cell membrane is the nucleus. And between the cell membrane and this nuclear membrane, this is the nucleus, and between these two membranes, whatever is present inside this area, including the nucleus, this is called the cytoplasm of the cell. So how can we determine that a cell is producing some kind of proteins, like the proteins which makes our nails, our hairs, our muscles? So how do we know that these very cells are capable of producing these very proteins? And obviously, if we want to observe the proteins, we will have to extract the proteins. And to extract the proteins from this tiny cell, we need a technique which is called the cell fractionation. And in cell fractionation, you will define the cell fractionation as that takes cells apart and separates the major organelles and other subcellular structures from one another. So as you see, here are the cell organelles, like the nucleus is the cell organelle, and as you notice that cell is very much microscopic, we cannot observe it with the naked eye. So how can we extract or get these organelles, which will be much more smaller than even the cell themselves? So for this very technique, for this very purpose, we use cell fractionation. And what we do in this process is we take tissue which is composed of same types of cells, and like the skin cells, and then we take those cells to put them into the test tube like this. And these are the tissue cells, as you can see in this test tube. Then we put this test tube into this mixer or grinder. Why? Because we want to homogenize, means we will put some enzymes in it, or to get the suspension, we will homogenize it, and homogeneous mass of cells will get through this very process. It is based on the size and density of the cell's content, like the cell organelles. Like the nucleus, it is larger than microsomes and ribosomes and mitochondria. These are the organelles which are present. Peroxisomes, these are the organelles which are present, but we will separate them, or this very mixer will mix all the things, like it will tear apart its cell membrane as well as the nuclear membrane, and after tearing apart, this cytoplasm is exposed and the organelles are also exposed. So in this very test tube here, you will get a homogenate. A homogenate is a mixture in which different cellular organelles are, we can see the cell debris which we mixed, right? We will get the homogenate, and then after homogenization, we will spin it through the centrifuge machine. As you can see here, these are the two beakers and this is the centrifuge which we set for a certain RPM, and we get different organelles on the basis of their size and density. So that's why it is called differential centrifugation. Like the heavier are the particles or the organelles, they will be settled down first, and then so on and so forth. Like the other organelles which are having less density and which size is smaller than the larger one, like the nucleus and the mitochondria, they will settle down to the bottom after an hour or so. So this is the process. You will do the centrifugation process. This is differential centrifugation because we get different organelles on their size and density based when we spin it through different RPMs. Like you can see here, as you can see here, the upper part which you can see here is the supernatant. This is the supernatant position, and after this centrifugation, the denser the particles are, they will settle down first. Like the nucleus, you will get it first. If you can see here, it is the 1000 G's. Like if you spin the centrifuge machine up to 1000 RPMs, revolution per minute, at this very position, you will get the pellet, which is the pellet. This is the pellet, and this is the leftover, which is in the upper portion of the test tube, is called the supernatant. So in the pellet, when you spin it at 1000 G's, you will get the pellet which will be rich in the nuclei, like the nucleuses, which are larger organelles, and cellular debris. You will get it at the 1000 G's. And then you will take this supernatant and pour it down into another test tube, and then you will repeat this process again. We will centrifuge it again, but with the different RPM, like the 20,000 G's for 20 minutes. For this, for 1000 G's you did it for 10 minutes, and for 20,000 G's you will do it for 20 minutes. After 20 minutes, you will see that the pellet will be rich in mitochondria and chloroplast. So this way you can see that the more you spin this test tube, or the more you increase the G's on this test tube, the more time you spend on the test tube, the more weight it gains, and you will get the pellet which will be of mitochondria and chloroplast. And when you increase it to the 80,000 G's, 60 minutes, for 60 minutes, 80,000 G's, and you will see that for this very much time, you will get the pellet which will be of the microsomes, which will be the tiniest particles inside the cells, and pieces of plasma membranes. Like this is the plasma membrane. You will see some of its pieces which will be composed of lipids and proteins, some carbohydrates maybe. And cells internal membranes, like mitochondria do have a membrane, it's a double membrane bounded structure. Chloroplast do have a membrane, it has a double membrane bounded structure. So this way you can get different organelles on the basis of their densities and size. And when you increase the span into 150,000 Gs for 3 hours, you will see that the ribosomes, if you want to get the ribosomes through this process, you will have to set the centrifuge, this differential centrifugation on 150,000 Gs, and only then you will see the ribosomes. If you want to get the ribosomes, you will have to set the centrifuge machine to 150,000 Gs, and then you will get the ribosomes. Otherwise, if you span it at 80,000, 20,000, 10,000, these ribosomes are too tiny that you cannot get it properly. So this way, this cell fractionation method, molecular biologists or biochemists use this process in order to get the different molecular weight proteins and other chemicals which are having denser or heavier than the rest of the molecules. You will get it. And if they are smaller, like the ribosomes, you can increase the centrifugation RPM, and you can get the different contents which are present inside the cell. This way you can examine that why certain cells are producing cancerous chemicals which are leading, or which are the cause of cancer, and the oncologist do this process to collect those materials from the cells and then examine them, and to know them well, that what are the elements which make them and how we can stop them or reduce them. So this is a very best technique. Hope this makes sense to you. Inshallah, see you in our next video. Take care. Allah Hafiz.